but we should get started with our next invited talk by Jenny Byrne of New South Wales Pathology and the University of Sydney. She's agreed to talk to us about enhancing the self-correction capacity of science through improved reporting and flagging of published errors. And I want to remind you that if you have questions, we may have time for a few questions at the end of the session before we go to the lightning talks. You can indicate your question by typing them into the Q&A box. Jenny.
Oh, Alex, thank you. Thank you very much, and to Jason for the invitation to speak today and all of the AIMOS committee. As Alex mentioned, I'm with the statewide biobank here in Sydney and also the Faculty of Medicine at the University of Sydney.
We did lose your video, Jenny. I don't know if that was intentional because I remember you had it on before.
Yeah, sorry. Look, I'm just sorry. I'm too nervous. I'm so relieved to actually be here showing my slides. I don't think I can do the two. I hope I'll put my video on afterwards if that's all right. Yeah, so look, I'm just going to talk about an issue that I guess I've seen through reading the literature over the past years, and that is the need to reduce the publication of errors and improve our capacity to correct them. And I think at the moment, you know, this mismatch between published errors and correction capacity is an issue. Essentially, if we had a situation where we had very few published errors, we could deal with a very limited or inefficient capacity to correct them. But if you have many published errors, essentially you really need a very efficient and scalable capacity to correct these errors. So both of these situations are kind of okay. But I think what we actually have is a situation where we have many published errors combined with a very limited or inefficient capacity to correct those errors. So, and certainly from my experience in reading the literature that I'll be talking about today, many overarching assumptions about error correction are essentially incorrect, and that is that published errors are infrequent, that peer review is effective in terms of detecting errors. I mean, there is literature to show that's not the case. The assumption that researchers will detect published errors after, you know, they're published, and that at the end of all of this, there are effective methods to correct the literature. So what have we proposed? Essentially, we propose that there is unfortunately a system of mass or industrial scale research fraud, which we are concerned may be operating through organisations such as paper mills. And I think Sandy made a reference to paper mills in his talk. We're concerned that there are fraudulent or fabricated papers that claim to examine particular human genes and human cancer models to shed light on the roles of those genes in cancer. We think that paper mills use templates, which gives rise to a sort of theme and variation look to these papers for speed and efficiency. But inevitably, rapid content generation is likely to produce higher error rates, and that's what we believe that we're studying. So these are the first examples of papers that I actually stumbled upon, and you can see that the titles of these two papers, which I should say are retracted now, include a gene name, contributing to a cellular property in a cancer type. So you can see that in both of these cases. Gene, proliferation, gastric cancer here. 3 other papers, again, gene, process, oral cancer, gene, liver cancer, process, etc. So 2 out of these 3 papers are retracted as well now. These papers showed many striking similarities, including the figures. So this is figure 2 in one of the papers. This is figure 2 in the second paper, published by completely different author teams. And I started studying these papers in detail because they're all examining the same human gene. Actually, I cloned that gene about 20 years ago. And if you're studying genes, you need to use small pieces of DNA or what we call nucleotide sequence reagents to do that. These sequences, as I'll show you, are very repetitive. You can't read their sense by eye. They are a language in their own right. And so to tell people what they actually mean in a paper, you need to link them to a text description, something that a human can read. And it's these links between sequences and their descriptors that can actually be verified. This is one of the papers that I showed you before. So here are 2 nucleotide sequences within that paper linked to 2 different identifiers. The only issue here is that this is exactly the same sequence that's described in 2 different ways. And the data show that using this identical sequence in supposedly different ways gives rise to a different result. That's not possible from a scientific point of view. So we described 48 of these papers in Scientometrics in 2017 with my colleague Sarah Labbe, and we then proposed in more detail what we were concerned might be happening, and that is that out of the thousands of understudied genes that exist in the literature for humans, that they could be selected, and then multiple manuscripts built around each of those genes. And we proposed ways that those manuscripts might be assembled, features of the manuscripts, and ways that these manuscripts or the creators of these manuscripts might take to deliberately evade detection. So this idea is not something that we've come up with independently. There has been, increasing discussion about the possibility of authors using external agencies or paper mills to publish. And this is an example of a paper published in 2016, which talks about the pressure, for example, of hospital doctors really needing papers and needing to take effective methods to produce them. I think Sandy referred to this as, this is what we need to survive, leave us alone. So when we examined these gene papers, we found that there are essentially 3 different nucleotide sequence identity types. So essentially we can have a reagent that's claimed to target gene X, but actually targets a different gene. So that will generate irrelevant or impossible data. You can also have a reagent that's claimed not to target anything, that's just meant to be a negative control. Instead it targets a gene, so that's clearly an invalid negative control. Or if we have a gene reagent that's supposed to target a gene and it doesn't target anything, essentially those data become impossible. So Sarah Labbe in France created the Seek Blastn program, and we published that in 2019. So this program can scan text, look for features and nucleotide sequences, extract their associated claimed use, and then we can verify that using an independent program called Blastn, which can then assemble a new list of facts that can be compared to determine whether or not the claim status is supported by independent analysis. We published that in 2019 in PLOS ONE, and this tool is freely available. So we've been communicating papers with these kinds of incorrect nucleotide sequence reagents to journals since 2015. We've literally communicated hundreds of these. Many of these notifications have not produced any published responses, to the point where at this point I feel that communicating more papers seems, not to be the best use of our time. So actually during lockdown, I decided to do something that I'd wanted to do for a long time, which was to analyze journal responses to human gene papers with a specific error type. And we chose to just analyze papers that have got this non-targeting reagent that instead is predicted to target a human gene. So just to highlight, that's this particular error type. These reagents are not meant to target any gene, and yet we can indicate that they are targeting something. So here are the 2 reagents that we reported in 2017. We just gave them nicknames, sequence A and sequence D. You can see they're just lists of letters, but when we apply Blastn to those, we can identify identical regions of sequence that are precisely corresponding to a human gene, indicating that they are likely to be targeting that gene. So we focused on these particular papers because most of the papers that we described in 2017 did have this kind of error. It is also a fatal error, because in these gene knockdown papers, every experiment is paired with the non-targeting control. And we felt that by focusing in on these papers, we could compare journal responses to what are very similar papers. These papers are all using the same experimental technique of gene knockdown. They're all using very similar model systems, and they all have this same fatal error type. So when we looked for journal responses, we found 31 human gene knockdown papers, most of which we had communicated. One other was identified independently by the journal, and one other was communicated independently by the authors. And when we looked at the responses, we found that this generated 14 retractions, 5 expressions of concern, 7 author corrections, and 6 decisions where the journal told us that they had decided to take no action. So importantly, this is very different from a journal just not doing anything. This is a journal saying, we are not going to do anything about these papers. Those of you with sharp eyes will realize that these numbers add to 32, and that's because one expression of concern eventually resolved into an author correction. So clearly we have a range of responses to a set of very similar papers. We noticed that there were clearly different responses to the same error, but including by individual journals. Some individual journals would retract a paper and then issue corrections. We had temporary slow responses, as I'll show. We had responses that did not refer to the error that we reported, and of course the journal that chose to take no action. So this is a timeline of these responses, and we have the different papers that we reported these papers to here on the left in different colours. The solid lines are the retractions. Sort of these large dots are the expressions of concern. The smaller dots are corrections, and these papers are the ones where the journal took no action. So you can see that, you know, there's varying lengths of these lines. We have a retraction that was very quick, within 3 months. We have some retractions that took nearly 3 years. And of course, what's not shown here is, of course, all of the papers where we're still waiting for a response. So when we analysed some of these published notices in detail, we noticed inconsistent explanations for what was an identical control from the same supplier. For example, here we have 6 author corrections where the supplier stated that they just provided the wrong sequence and they've now provided the right one. We had another correction where the author stated that the negative control is wrong, but that this doesn't matter. We had retractions where authors stated that the negative control was wrong, where the authors couldn't confirm the use of the corrected control sequence here, and other kinds of explanations. And essentially, collectively, these explanations are not all possible for the same reagent from the same supplier, and so some of this information may not be genuine. In fact, many factors should actually facilitate the correction of these incorrect non-targeting controls. We reported them in early 2017. There are many of these papers, and many are visible very easily in the literature. There are multiple companies that claim to have supplied these incorrect non-targeting controls, and indeed we have 2 published corrections where a particular company accepted responsibility. So given this, it's very unusual that there are so few published author corrections. For example, the 2 pink lines here are the corrections where the company accepted responsibility. This happened in October 2018, and yet, you know, literally years later, we have outstanding expressions of concern that could have been corrected but have not been. So to summarise our concerns, this analysis of journal responses to very similar papers shows that there's a very thin line between issuing a retraction, an expression of concern, a correction, and indeed doing nothing at all. There were inconsistent explanations for the same reagent supplied by a single company, and an apparent lack of interest in correcting these papers, given the opportunity that exists to correct them. So that's a lot to unpack, but I think when we thought about what we need to do, we also thought about what we could have done better. We certainly made our own mistakes. One of my favourites was reporting a paper to the wrong journal, and I've done that at least twice. That clearly slows things down. But we reported errors as well by email, and we started to think about this approach and realising that perhaps it's actually challenging to convey information by email, particularly about complicated things like non-targeting controls. Information might have been missing as we follow up, and the email threads get longer, information is diluted. We had trouble knowing who to contact, and we also found evidence of editorial staff turnover, particularly over periods of time in excess of 1 year. So what we're proposing, and this is really not rocket science, is that there is a standardized error reporting template that journals and publishers should largely design so that we get the information that they need in the format that they want. There's then a defined communication pathway which can translate to a commitment to follow up. And this error reporting template could be shared between authors and third parties. It could be posted at PubPeer. It could eventually be published and be a starting point for further actions. And in work that is currently in revision, we have a draft of this template. It really doesn't matter what it looks like. It just matters that it's going to work for everybody. And we have proposed that this error reporting template could align with the fields that were proposed in the REAPPRAISED checklist published by Andrew Gay and colleagues in Nature earlier this year. If we're going to have better methods of reporting, of actually flagging papers, particularly with errors such as verifiable errors. And early this year, we published the use of something that we've called an editorial note, which I've described as just running a yellow highlighter across the paper. We hope that this would be faster and less loaded than an expression of concern, and by being published more rapidly, it would provide an earlier starting point, allow the transparency of publication investigation status to be disclosed. This might also hasten investigations and promote resolution. So just to really quickly compare editorial notes and expressions of concern, deliberately the tone of the wording in an editorial note would be neutral. I think one of the issues with expressions of concern is indeed that word concern. That can delay publications of expressions of concern. In our experience, these are slow, and that is also mirrored in the literature around expressions of concern. We would only expect editorial notes to ever be a placeholder. This is intended for expressions of concern, but as I've just given a few examples, expressions of concern often persist in the literature somewhat like a light form of attraction. So in summary, if we had access to clear standardized error reporting templates and rapid and transparent error flagging through something like an editorial note, we might achieve more frequent, timely and accurate error correction. And we might move away from the current situation where we have many errors and a very limited capacity to correct them, to a system where really it's not so important whether you have very few errors or many errors. If you have an efficient and scalable correction capacity, you'll be able to remove these from the literature. I think in addition, we really need a better evidence base to support error detection and correction. We need to understand how frequent are published errors in different fields. We need to understand how better peer review can better detect unpublished errors before they enter the literature. We want to help readers to detect published errors, and we need to understand how error correction can be improved. Do changes actually make any difference? So we have published further discussion around issues around possible systematic research fraud in the human gene literature, and more recently, we've provided a how-to guide as to how journals and peer reviewers might be able to detect manuscripts and publications that might have been produced with the assistance of paper mills. Above all, despite this being a very unpleasant topic, we do need to talk about systematic fraud, because by ignoring it, it is not going to go away. So I'd just like to thank my colleagues, my many colleagues in this work, notably Yasunori Park, who has helped me a great deal with the present manuscript that I've presented today, and of course Cyril Labbe for writing Seek Blastn. I want to thank the NHMRC for funding that's supporting our current research. And just before I go, I really want to tell you how excited I am about Associate Professor Andrew Gray, who will be presenting at the Biobank in a kind of post-AIMOS event on Monday, the 7th of December. It's a free talk, and I'll share the link in Slack. So Andrew Gray was behind the publication integrity checklist that I mentioned. Thanks so much for your interest. I'm very happy to take questions.
Thank you, Jenny. Wait for the applause to die down here in the Nerve Center. But we don't have any questions quite yet in the Q&A, but I wanted to ask, what's your thoughts about how, in my experience, journals are slow to do anything, let alone, I mean, even things that more directly serve what they perceive to be their interest, such as publishing papers, which is different from admitting that they let some things through that perhaps they shouldn't have. And so I wonder what you think about things like PubPeer and other commenting services to remove the bottleneck of the journal from the equation.
Thanks, Alex. Yeah, look, I think PubPeer is it's a very valuable addition to that sort of error collection landscape. I guess one of the things that I know from talking to my colleagues is that some people use PubPeer and not everybody does. And I think that everybody uses the literature. So I think what we need is more of a sort of a cross-fertilization between platforms, so that comments, end up somewhere like PubMed, which is basically where researchers read them. I know PubMed used to have a comment function, and we've actually used that in our research, but then they unfortunately they stopped supporting that through apparent lack of interest. So yeah, I think PubPeer is part of the solution, but we need to have more corrections actually coming into the literature where everybody reads them.
Adrian Barnett, you may know from Queensland, says, thanks, Jenny. One other thing we need is more funding for this kind of work.
Yeah, no, that's very true. And actually, I was asked. I actually asked a question to Paul, but I think I was in a different platform, and my questions might not have been coming through. But you know, my own view is, I think, like, I actually got an ideas grant to study this, which is quite extraordinary. But and somebody did say to me, that's never going to happen again. But I kind of think we need different pools of money. You know, we need pools of money for replication studies. We need pools of money for meta research, because sometimes it can be difficult, particularly in the health sciences, to compete with people that are actually saying, you know, they're going to cure cancer or they're going to create, you know, a new treatment, which is of course fantastic research, but all of this relies upon the integrity of the literature. And if we don't have this same investment in ensuring that the literature's at least as good as it can be, you know, a lot of these other efforts are going to be very difficult.
Yeah, I saw that in Paul's slide. It was great to see that the NHMRC had announced what, I don't know if they called them principles, or including things like replication. I don't know if error detection was in there, but I don't know whether we're seeing actual funding specific to those things. And as you say, maybe it does need to be specific. Otherwise, we can't beat out the groundbreaking work. It sometimes breaks things more than sets a foundation for things. If anybody else has any questions, they could type into the Q&A. Oh, I see, I believe Dom has written in on the chat, Dom Roche. He says, what are your thoughts on the work being done by Retraction Watch? Could they do things better to help correct errors in addition to documenting them?
Yeah, look, I think Retraction Watch does a really great job, but I think their role is largely to talk about, you know, they maintain a database of retracted articles, which people can use for their own research. But at the end of the day, you know, it's the journals that are publishing these papers, and so if they're going to be corrected, that needs to happen through the journal. So I think, you know, organisations such as Retraction Watch are invaluable, but, you know, there are probably 30,000 biomedical journals, and they, as a collective, have tremendous capacity to, you know, correct the literature. And I think that's why we've thought about this sort of reporting template, because clearly, you know, they need to tell us how this needs to work from their perspective. You know, a lot of the time we write to the journals and we say, you know, we found this, I'm happy about this, etc. But I'm not sure we're always telling them what they need to know or what they want to know. So by allowing them to lead the conversation, hopefully we'll have a better conversation.
An currently anonymous attendee asks, what can PhD students and early career researchers do to help move this kind of work further?
Now, that's a great question. I think, you know, read the literature really carefully. You know, analyze papers. Don't necessarily trust what you read. If you're a genetics researcher and you're thinking about, you know, using nucleotide sequence reagents in your own work, I'd really advise you to cross-check them. You know, use tools that exist to validate published claims. It's actually not that hard. It might take a couple of minutes to search a couple of DNA sequences, but you can literally save yourself months or years of time in the laboratory. So, yeah, read the literature with a critical mindset.
We have time for a couple more questions. I was wondering, trying to think about when we've seen the most success historically in some kind of reform that took place across many journals. And I think of the ICMJE, the International Committee of Medical Journal Editors, and Paul alluded to how in his talk, must be 20, 30 years ago now, that they started requiring that clinical trials be registered, I believe, for their member journals for them to be published. And so I suppose we need something like that again, don't we, where a whole bunch of journals together agree on a way forward, like with your framework.
Yeah, that's another thing that we proposed. We unfortunately, you know, sort of know from the science behind the scenes that a tactic that paper mills can take is just submitting manuscripts to many different journals at once and seeing where they stick. And a way of stopping that would be essentially mandating preprints. So, you know, duplicate preprints are very easily detected. So I think if journals were to rely more on preprints, then at least those kinds of duplicate submissions would be deterred. That's one way. But I agree, you know, journals getting together and agreeing that you know, we're going to prioritize post-publication reports through some kind of standardized reporting system. I think we have.
Jason has jumped in with multiple questions. He's got one which is, thanks, Jenny, incredible stuff. What can we do to incentivize people to conduct more error detection and correction work?
Yeah, I guess that's just sort of funding and support, you know, recognizing the field as a field in its own right. Yeah.
And he puts on his lawyer hat perhaps when he asks, are there any legal redresses if a fraudulent paper causes harm? Oh, he says that's kind of been answered, but I missed the answer.
Yeah, I don't know. I'd be looking to Jason for his legal mind there. And it's a good question. I don't know. Yeah, I mean, certainly there are cases where fraudulent papers have caused harm. There's many cases of that in medicine, unfortunately.
Jason, do you want to speak to that about, you mentioned negligence tort law?
Well, I guess one defense is the truth, but they don't have the truth in their favor, so I would think they couldn't use that. I don't know. I was just spitballing, Alex. Sorry.
We've seen things go the other direction, haven't we, where articles that have been retracted or threatened to be retracted, one of the authors has sued. I don't know who they sued, but whether they sued the journal or whether they sued their accusers.
Yeah, I think that is something that obviously, you know, is a cause for concern. And I suppose, you know, I'm certainly not insinuating that retraction should be undertaken lightly, but I think, you know, just what we're showing, what our experience is that the system it's not working. And I think what I haven't shown today is, you know, the many papers that we've reported, you know, as early as 2015, but many of which were reported in 2017, where we're still waiting for something to happen. You know, it's just too slow. And there are some journals that clearly just don't prioritize this, and I don't think that's right. I don't think that journals should be indexed in places like PubMed if they're not committed to ensuring quality.